Unmute. শুনতে কেন পাচ্ছি না?
Welcome, everyone, to the Her Story of Science Celebrating Women in Research conference, organized by Biologically Speaking in association with BiasWatch India. Biologically Speaking is a series started by a group of doctoral students and postdoctoral fellows from USA, India, and Europe across different time zones. We established in 2021. It is a new team, but we are a bunch of enthusiastic individuals exploring the wonders of the biological world ourselves and hoping to bring you all on this ride with us too. Take this fascinating journey through the nooks and crannies of the bio world. Coming to Her Story of Science conference, To commemorate the International Women's Day of 2021, we bring to you this 2-day conference consisting of 7 sessions each dedicated to a woman scientist. And additionally, we have 2 keynote lectures. We are joined by participants from around 17 countries and 500 participants. So thank you all for joining us. We are really overwhelmed by your support. We might be new, but your support means a lot to us. We are truly inspired by all our speakers for this conference, and this is our small endeavor to celebrate the exemplary work done by women scientists in India. So to show our gratitude, before we begin, we would want to share a small video with everyone. Hope you enjoyed our little video. We have really worked behind this. This has been our dream child, and we are so happy that we have been able to receive the support of so many wonderful speakers and also our audience members. In addition to our team, who are all different doctoral students and postdoctoral fellows from different parts of the world. We also want to give a huge shout out to our outreach ambassadors, Vani Pandey from ISER Pune and Debayan Banik from Amity University, Kolkata. We'll start our first session in a few minutes at 10:30, which is the scheduled time. So we'll wait a few minutes and then we'll get started with our first session. Thank you all for being here. I would like to also take this opportunity to mention that as our effort to disseminate scientific news from all over the world and the journeys of different scientists, we also have monthly webinars on the Biologically Speaking Forum. So please follow us on social media and follow our website to join us for our webinars. We do have speakers from different backgrounds, different research interests, and we'd really love for you to join us. With that, I would like to introduce our first session. As I mentioned before, that each of our sessions are dedicated to women scientists. Our first session on biodiversity and animal behavior is dedicated to Jagannathan Vijaya. Jagannathan Vijaya was India's first female herpetologist and was known as the Turtle Girl for her immense contribution to the conservation of turtles. She documented the movement of turtles all over the country and worked as an assistant to Edward Moll, the chairman of the World Conservation Union's Freshwater Chelonian Specialist Group for his nationwide surveys. J. Vijaya, or Viji, as she was fondly called, was born in Bangalore in 1959. Since childhood, she was an avid admirer of nature and grew up idolizing the likes of Jane Goodall, Birud Kalkidas, Jacques Cousteau, Dan Fossey, and Reinhold Messner. She started working with Edward Moll at the age of 22 and documented the entire trade line of certain species of turtles, right from their capture from the habitat till the transport and selling in the meat markets. She was a champion for captive turtles and played a pivotal role in freeing them from captivity and ending the illegal trade. So in honor of India's first female herpetologist, Biologically Speaking is proud to present the Jagannathan Vijaya session on biodiversity and animal behavior. For this session, we have three speakers, Dr. Anindita Bhadro, Dr. TNC Vidya, and Dr. Uma Ramakrishnan. Our first speaker for this session, I'm absolutely delighted to introduce her, Dr. Anindita Bhadro from the ISER Kolkata Institute. Dr. Anindita Bhadro is a behavioral biologist and an associate professor in the Department of Biological Sciences at Indian Institute of Science, Education and Research, Kolkata. Her lab works on the behavioral ecology of free-ranging dogs in India to understand the evolution of dog-human relationship. For her PhD work, she worked on the social behavior of the paper wasp, Rupalidia marginata, at the Center of Ecological Sciences, Indian Institute of Science, Bangalore, under the guidance of Professor Raghavendra Gadadkar. She was awarded the INSA Scientist Award for her work. Later, she received the SERB, or Science and Engineering Research Board, a Women Excellence Award by Department of Science and Technology, and the IAP's Young Scientist Award. Dr. Bhadro was recently awarded the Janaki Amal National Women Bioscientist Award 2020-2021. She is also the founding chair of the Indian National Young Academy of Science and the co-chair of the Global Young Academy. Her work has been reviewed in Nature India, Science News, BBC Earth, and Discovery News. And in addition to that, she's involved in outreach work. And it's also interesting to know that she has a theater group called Mukosh. So besides her research, she does wear other hats. We are extremely happy to have you here, Dr. Bhadro. Thank you for joining us on this session.
Thank you, Sanjana, for the nice introduction and for the invitation to this fantastic meeting. I mean, the idea itself is so nice, and you have a great lineup of speakers. So it's a pleasure to be here, and I'm really happy to be, part of the great session with very nice speakers. So, shall I start?
Yes, absolutely. If you,
Okay,
yeah, you would be able to share your screen. So if you want to share.
right. Yeah, can you see my screen?
Yes, yes. Perfect. Thank you so much.
Fine. So I'll just keep the video off while I speak, if that's okay.
Yeah, whatever you want. Yeah, absolutely.
Okay. Sometimes otherwise there's a bandwidth issue. Um, Good morning, everyone. And it's a pleasure, as I say, to be here. And I'm really amazed to see the effort that you have put in for getting this meeting together. So I will be speaking briefly about part of the work that we do on free-ranging dogs in India. And at the end, I'm happy to take questions. So when I got this invitation to speak, I thought of, which part of the work should I be speaking on and what aspects, because often I speak on the dog-human interactions part, because that is what people find very fascinating. But looking at the title of the conference and looking at the kind of, background, I decided to speak on something slightly different this time. So since this is her story of science, I, did a quick search on Google Images to see what you get when you search for something like work-life balance. And when you search for work-life balance for women, you get images like these. These are the top images that come up on Google Images. And as you can see, these are all women who are trying to juggle multiple responsibilities and tasks while they are also having a career. That is the idea behind these images. When you do the same search for men, you get something like this. I was quite intrigued to note that for men, life denoted music, hobbies, wine, a relationship perhaps, and then there was work. But when you looked at women, there was inevitably a baby somewhere. And this was quite intriguing because, of course, we understand that women, when they are at a very critical age, they also have the time when they can choose to be mothers, and motherhood obviously gives us an added responsibility and makes life difficult to balance, because you have this very, very demanding human who you need to take care of. And of course, because of biological reasons, the responsibility of a baby falls more on the mother than on the father, even though the father might want to help. And this is where I thought I will talk about my work on motherhood, not in humans, but in dogs. And I will try to give you a slightly, detailed overview of some of the work that we have done to understand motherhood in dogs. So in behavior, just to give a very brief overview, um, people like us try to understand behavior using proximate and ultimate questions. And this framework that you see out on the slide was given by Nicholas Tinbergen, one of the three Nobel laureates who were the only people ever to receive a Nobel Prize for working on ethology or behavior in 1973. So Tinbergen's four questions, as this is popularly known, is broken down into how a behavior occurs in an individual, how the individual how it occurs during the lifetime of the individual, which is the ontogeny. Then it is why the behavior is there in the species, how does it help the species, what is the adaptive value of the behavior, and What is the phylogenetic history of this behavior? So the evolutionary paradigm. So we behavioral biologists typically try to understand how an animal lives and why it lives the way it does. And I will try to give you glimpses into some of the questions that we addressed pertaining to motherhood, both in terms of proximate and ultimate questions. So if you look at a pup's life, when a pup is born, you see that it is part of a very complex network. And this network here is created only using play interactions. So obviously you understand, if you look at more interactions, it becomes more and more complex. So in this network here, the light blue circles denote pups and of one mother. And then there are the dark blue ones, which are other puppies in the group. And then there is the mother in red, and I will come back to this, allo mother and putative father and humans. So the pups are interacting with all kinds of individuals in their lifetime. But of course, when they begin their life, the most important individual in their life is the mother. So we went ahead and studied parental care or maternal care, and we began with how does the mother show care, what are the various forms of care, and what are the stages in the pup's life that the mother gives care to the pup. We found, after looking at a very, very large number of mothers, that they selectively choose certain sites as the place to give birth. So this is called the den. Dogs don't really have fixed shelters. Otherwise, they just, have their favorite spots where they sit and rest or lie down and sleep, but they don't have dens. They have dens only when they give birth. And dens are of all kinds. You see some images here. They can be in somebody's courtyard. Kinds of dens that we found. This was data from 105 dens. And what we eventually ended up finding is that mothers, they spend a lot of time and energy to search for dens. They keep rejecting sites until they find one place which is suitable for them, where they give birth. And these dens, unlike other animals in urban habitats where they try to go away from humans and hide their babies from humans, in case of dogs, they actually seek out humans. So they try to give birth close to humans, and they also prefer to beg from humans when they have small babies, and human given food is their main source of sustenance when they have little pups. So this is a very unique feature of dogs, unlike all other animals which live in proximity with humans. When we looked at the various kinds of caregiving behaviors in mothers, obviously the dark yellow that you see here denotes suckling or nursing. And obviously when the pups are very small, then a mother has to spend a lot of time nursing them. So producing milk, letting the pup suckle, and that takes up a lot of time and energy. Often you would see the mothers are visibly losing weight. You can see their ribs coming out because a lot of energy of reserves of their body is being spent. Other than that, the mother is cleaning the pups, cleaning the dens, protecting the pups, playing with them, all kinds of behaviors. And a very important behavior, the pups are typically born in the winter. So the mother will curl around the pups and they will sleep in a pile, which obviously helps in thermoregulation. So there are a numerous amount of behaviors, and we found that as the pups are growing older, the mother is budgeting her time allocation in these different behaviors in different manners. Some behaviors are taking up more time when the pups are smaller. Some behaviors are taking up more time when the pups are older. For example, protective behavior. When we looked at how much care a pup receives, because in a litter, there can be one pup, there can be 15 pups. So from the perspective of the pup, this graph here denotes, you see age of pups in weeks on the x-axis. You can see the size of the litter, which is increasing in this direction, and the proportion of active care which a pup receives in the litter. So the total care a mother provides is divided by the number of pups that she is caring for. And what we saw here is obviously that the litter sizes, when they are small, there the individual level care for a pup increases as compared to when the litter is larger, which basically means that there is sibling rivalry. Because if there are a lot of puppies, then among the pups, the care gets distributed because the mother does not have infinite resources. She cannot keep scaling up her care per pup as the pups grow older. So there is regulation of active care to some extent, but after a certain level, the mother saturates her care, and then there is competition between the pups. So for the pup's perspective, it is good if there are lesser siblings. But of course, from the mother's perspective, it is better if she has more siblings because some would die and some would survive. When we looked at the most important maternal care behavior, which is nursing, we saw that as the pups grow older, nursing steadily decreases, or the time spent in nursing steadily decreases. And there is a point of time when the pups are 14, 15, 16 weeks old when nursing completely stops. Then the pups are weaned and then they become independent of the mother's milk and they become. This was interesting because we often you expect that she would be nursing at a steady rate and then suddenly stopping, but this is not happening. So we wanted to also look into this weaning behavior, as it is called, in a little more detail. And I will show you this clip here. This is not playing. I don't know.
Is a video not playing?
Yeah, it's not playing. Okay, because of the highlighter. So this is something that many of you would have actually noticed on streets. And when the mothers do this, often people find it very difficult to accept. Then we have even seen people grabbing the mother and making her stand in one place so that the pups can nurse from her. But this is weaning and it is part of the life of the dogs and of any other animal for that matter. So we looked at individual bouts of suckling or nursing. This graph here shows the proportion of suckling initiated by pups in case of the red triangles and mothers in case of the open circles. If you see here in the 3rd, 4th, and 5th weeks, the proportion of suckling bouts initiated by mothers and pups are almost equal. So both the mother and the pup are equally interested in the act of nursing. 6th week onwards, the mother's interest goes down and the pup's interests are high. And as you see here, from 7th week onwards, 100% of the bouts are initiated by the pups. So the pups are trying to suckle, whereas the mother is trying to refuse as soon as possible. And this window of 8 to 13 weeks, when we see this discrepancy of 0% and 100% is denoted as the weaning period. This was very interesting for us because this reminded us of a very famous theory in evolutionary biology known as parent-offspring conflict theory, which was proposed by Robert Trivers way back in 1974 for humans. Trivers basically said that mothers and their offspring will have a period of conflict over weaning, one time when the mothers would want to wean and the pups or the offspring would want to continue to suckle. After a point, both parties will agree that weaning is necessary. And this is based on the idea of genetic relatedness and reproductive success. And I'm not going to go into the detail of the theory. But what we also looked at was the duration of each bout. And what we found was the red bars are for the mothers and the blue bars are for the pups. What we found was in the initial stages, when the mother and pup are in agreement, then the bouts are quite high when the mother is initiating the suckling. However, after the 7th week, you see there are no red bars. So mother-initiated suckling has dropped down to 0, and there are shorter bouts of nursing because mother is refusing to nurse more and more often. She is terminating the suckling bouts quite faster, even when the pups are initiating them. And this window completely matches with the window that I showed you earlier. So we said this looks like that 8th to 13th week is the period of weaning in dogs. And this is important because if somebody is going to take a dog as a pet, then this is the period when they can target to take the dog away from the mother, not before that, when they need to be with the mother. But of course, this period is also when they're learning other things from the mother. It is not only the milk that they're dependent on the mother for. So we created a timeline of maternal care. Up to 7 weeks, there is very high level of maternal care with a lot of suckling or nursing. 7 to 13th week is parent-offspring conflict. And beyond that, it's mostly passive maternal care. And after 16 weeks, they are becoming more independent of the mother. Now, this parent-offspring conflict window gave us the opportunity to do an experiment to see how selfish mothers can be. Selfish, of course, in evolutionary terms. So the idea was, is the mother going to be interested in her present litter, or is she generally interested in her lifetime reproductive success? And of course, when we say this, we are not saying that the mothers are sitting and calculating. These are obviously questions in evolutionary terms. So coming back to Trevor's theory, we said, okay, let us see. Of course, there is weaning happening, but then when it comes to sharing of food, because the mothers, as I said, are depending on humans more and more for food when they're having puppies. So when they are in this phase, when humans are more actively giving them food, because people say, oh, the poor mother, she has so many pups to feed. Let us give them some more rice. So this window of time, will the mother take this opportunity to feed herself and replenish her energies for the next litter, or will she feed the puppies more? This was the question that we asked, and we did a very simple experiment. So the two questions that we are addressing is that, is there mother-offspring conflict over extended parental care, in this case food sharing, and will the quality of resources available affect the cooperation level between the mother and the offspring? The videos that are playing here will show you two conditions. One, where the mother is actually sharing the food with her puppies, and the second, where she's actually competing with her puppies. So we did these experiments with 15 mothers and their offspring, or rather I should say we had 15 successful data points. We started with a lot more, but then most of the pups die in the middle and you cannot continue the experiment. And we finally got this data where we showed that the proportion of conflict is steadily increasing from the 8th to the 14th week, which is the period when we did this experiment. And not only is the mother showing more and more conflict or more and more snatching away of the food from her pups, but during this period, when we looked at the mother's body condition at 8 weeks, at 14 weeks, and at 11 weeks, we saw that she is definitely showing an improvement in her body condition. So the green bars are all at 8 weeks. So 8-week-old mothers with 8-week-old pups are all comparable in their body condition. So, a lot of scars, ribs showing, not very healthy fur. And at 14 weeks, which are all the brown bars, all the mothers are once again comparable, and there is statistical difference between the green and the brown. So not only is the mother showing a conflict, but she is actually using up this extra food for her own benefit. So obviously, the mothers are behaving in a quote-unquote selfish manner. But then we were giving them biscuits. The reviewers asked, what if you gave them meat? Maybe the mother will allow the puppies to eat the meat because that is protein. Puppies should not be eating biscuits anyway. So fine, we wanted to do this, and we took help from a physicist friend to model first what it would look like if the mothers were having selfish and altruistic strategies. So the line 1 here comes from the original data with biscuits, and line 2 denotes the two strategies where A, the mother is selfish, showing high levels of conflict even with from the beginning, and B, when the mother is altruistic, her conflict starts at a lower end and only gradually increases. And then we modeled this, then we went ahead and did the experiment. And here you see the video, only if you can see it. So again, we did this with a large number of mothers and their pups, but we ended up having, again, only some 16 successful data points. So here the mother is not even trying to snatch the food from the puppy, being a nice mom. Sorry. This one is my absolute favorite. Okay. So this was the model on the right, and here you have the data. In fact, it was such a close fit to the model that we were a bit worried that this is too good to be true. But anyway, we concluded that the mothers are behaving really, really selfishly when resources are higher, which means they are putting more importance on their lifetime reproductive success rather than on their immediate litter. So this makes a lot of evolutionary sense, and we were really thrilled with the result. And this was the first experimental demonstration of parent-offspring conflict in a natural system in a field setup. People have done a lot of models, people have done experiments in the laboratories, but a field data did not exist before this. Now I'll shift gears a bit and talk about what I like calling the Great Indian Giant families of free-ranging dogs. So we noticed that in a group, when there are multiple puppies, there are sometimes more than one female which have given birth. Of the 23 groups that we had data, 23 mothers that we had data on from 15 different groups, 5 received only maternal care. There were 3 which received care from another female in addition to their maternal care. So this was called female allo care, allo from a different female. 3 litters in which a male in the group showed care to the puppies in addition to maternal care, and 10 groups in which, or 10 litters in which they received all 3 kinds of care. And there was this one outlier, because here the mother died when the pups were 2 days old, and the grandmother adopted the pups, had a miscarriage herself, and acted like the mother, which was quite amazing. Who are these caring females? They are the maternal grandmother, they are older siblings of the puppies, and sometimes they are aunts or daughters of the same mother, who are showing care to each other's puppies. And what are these forms of care? They will come and lick and groom and protect, sometimes play, which made sense because there are all these pups staying together. And of course, if you have relatives giving care, this can have, again, very evolutionary significance. There is kin selection, and helping your relatives eventually helps you propagate your own genes to some level. So when we looked at female allocare in a little more detail, we found that obviously, as expected, the mother's care level was much higher than the aunts and the grandmother's care level, which again made sense. What did not make sense was we found that in some cases, the females, the related females who are showing allocare are not only just grooming and playing and protecting, but sometimes they are also nursing these non-filial pups. And we wanted to understand what was happening. But before that, let me tell you about the putative fathers, as we call them. We don't have genetic data. This is something we've been dying to do, but we don't have anyone who's interested in the problem like us, so we don't have collaborators. We found that there are males in the group sometimes who show care to the puppies. And when we looked at the types of care, here is an example. we found that the males are playing with the puppies, protecting them, sometimes even regurgitating food for them, as you will see in this video. The pups are trying to play, and the male is going to regurgitate food, which the pups are going to feed on. This was very interesting because often mothers will not allow males near their pups when they're very small, but then there would be this one specific male which would be not only allowed near the pups, but often the pups would be left in the care of the male when the mother goes foraging, and this male would spend a lot of time with the puppies. So we were very curious to find out the kind of care that the fathers, or the putative fathers as we call them, gave. So this was the graph that I showed you earlier about the distribution of time over different kinds of care by the mother. When we compared this with the father, we found that obviously the fathers cannot nurse, and there is a lot of play, which is in the black. There is a lot of protection, which is in pink. And when we looked at the data statistically, we basically found that while mothers are spending a lot of time feeding, fathers are spending a lot of time in play. In the sense that fathers play more than the mothers do, and obviously mothers feed more than the fathers do. So there is division of labor in a way when there are males who are taking care. Since we did not know whether these are actual fathers because the dogs are promiscuous, they meet with each other multiply, so we call them putative fathers. And when we looked at the suckling behavior of the mothers and the allo mothers, specifically we saw that allo mothers, who are all sitting here on the zero line, never initiated a nursing bout. So when we found pups nursing from allo mother, it was basically the pups who are grabbing the allo mothers whenever they find them. Pups don't care whether they are suckling from their mother or the grandmother or the aunt. They just want to feed, and they feed from whichever available lactating female. And this is why the allo mothers are seen to be nursing from time to time. And when we look at the termination, all nursing bouts from the allo mothers are always terminated by the allo mothers. Sometimes they will even bite the pup, like in here, if they find them nursing. So we found that the allo mothers are never nursing. They are basically the victims of milk theft. So the pups are receiving a lot of care from mothers, allo mothers, putative fathers, but then how are they surviving? Survival is very low. If you see this graph here, it shows pup survival and at various stages, and you see here the survival keeps dropping as they grow older, with the highest mortality occurring at the 4th month, which is also when they are becoming independent of the mothers and becoming juveniles. The highest is about 34% in a month. And the survival is highest at the 1st month when they are completely under the care of the mother. So the mothers, no matter how selfish they seem to be, are quite good at mothering. The causes of mortality, just to give you a glimpse, a lot of it is human influenced, and the actual data says that 81% of pups born in a season die, and 64% of the mortality is contributed by us. So we are the apex predators as far as dogs are concerned. So putting together all this information and some more which I have not spoken about, we have a very complex timeline of pup development with lowest mortality. And then when foraging starts and when they are in the protection of the den, the mortality is lowest. Then we have maternal and allomaternal care. We have parent-offspring conflict, we have milk theft. So very, very complex set of interactions going on within the dog group as the puppies are growing. So to conclude, a mother dog shows extensive care towards her pups, starting from den selection before the pups are born. There is limited levels of care. So the larger the litter, there is more and more competition between the pups. Weaning conflict is very real. Mothers begin competing with their pups over food during weaning. So competition is not only over the weaning, but also over food. Lactating females are often victims of milk theft. The mother-pup interactions also reveal interesting cooperation conflict dynamics, which might actually influence their behavior at a later stage in life. We have helping relatives and males, which add to the chances of survival of the pups. The free-ranging dogs, with all of this, suggest that they have a communal breeding system, which is completely unknown in canids before this. I will end by thanking my entire lab. So these are the images of my PhD and master's students here. And of course, there are always a batch of interns working with us, contributing to the work in the lab. And thank you to BiasWatch India, to Biologically Speaking for this great conference, all the funders, and Isaac Kolkata.
Thank you so much. We'll definitely want to give you a round of applause, but in a virtual setting, we have to make do with the reaction. So thank you very much. We do have a lot of questions. I'll try to take a few, and if you wouldn't mind answering the rest in the chat box, or you can, for participants, you can also get in touch with Dr. Bhadro later to ask her questions. So the first question is there any, so that this is from Somavali Dalvi, is there any difference between pups receiving only maternal care and pups receiving maternal care, allocare, and paternal care? Are there any long-term effects of this care difference?
Very good question, and the answer is we don't know. See, the point is the mortality is so high that it's very difficult to have pups which we can follow until adulthood. So this is something which is there in our long-term plan. We want to do this, but this work has not been continued after, because the students find it very tedious to do this kind of work. So I definitely want to do this and address this question, but right now we don't have an answer.
Okay, thank you so much. Another question is from Orpi De. Will the selfish behavior of conflict between the mother and the pup differ between street dogs and domesticated dogs, or is it uniform?
Domesticated dogs don't really need to do this in the sense that there is ample food and people regulate their behavior, right? People will not let them show conflict. They will feed the pups separately. So it would be interesting to check this out in pets, if people have pets with puppies. But my hunch is that this is so, intrinsic behavior, innate behavior, that given the chance, the mothers will show this.
Okay, thank you. We'll take one more question for now. It's from Shomona Kaur. How much time does a mother dog need to forget about her dead or lost pup?
A day or two.
Okay.
Appears to be. Of course, we cannot, probe into their heads, but we sometimes see them, whining away for a day or so, but then they kind of go back to normal.
Okay, we'll probably take one last question and then move on. So it's Malavika Menon asks, why is protective behavior of father high during week 14? I think it's she's referring to the graph that was there.
Yeah. So often what is happening is that this is the time after 12 weeks, the pups are also doing a lot of movement. And that is where we see that the mother is going away to forage for longer terms, and the father is, or the putative father is spending more time. And this is also when you would see that the pups are getting into fights with other adult dogs, and the putative father is showing more protection. So this is because of the dynamics happening around them.
Okay. Thank you so much. I hope people who ask questions, she was able to answer your questions, and others, I'm sorry, I wasn't able to get other questions. So please feel free to reach out to Dr. Bhadro for the questions. And with that, let's give her a round of applause. I mean, it's not very satisfying, but we'll make do with what we have here. And I would also request you to stay on for the rest of the session if you can. So
I actually have another talk to give.
Absolutely.
So I will leave in a minute or two. But yeah, it was nice, and I will get back some other time to the session.
Okay,
hopefully
thank you.
Thank
you so much. Thank you. And thank you everyone for staying with us. So for our session, the Jagannathan Vijaya session. We have our next speaker for this session. I'm very happy to introduce our second speaker for this session, Dr. TNC Vidya. TNC Vidya. Dr. TNC Vidya is a behavioral biologist, primarily interested in vertebrate behavior and socio-ecology, with broader interest in the areas of behavioral ecology, conservation biology, phylogeography, and evolutionary biology. She is an associate professor and the chair of the Evolutionary and Integrative Biology Unit at the Jawaharlal Nehru Center for Advanced Scientific Research in Bengaluru. The focal research of her lab is Asian elephant socio-ecology and behavior, carrying out long-term research in Nagarhole and Bandipur National Park, southern India. During her PhD, she worked on the population genetic structure and phylogeography of the Asian elephant at the Center for Ecological Sciences, Indian Institute of Science, Bengaluru, under the guidance of Professor R. Sukumar and in collaboration with Professor Don J. Melnick and Dr. Prithviraj Fernando at the then Center for Environmental Research and Conservation, Columbia University. Dr. Vidya received a postdoctoral fellowship from the National Research Foundation, South Africa. Her research there focused on mammalian sociogenetics at Stellenbosch University, where she examined the facultatively social yellow mongoose. She was the recipient of the Indian National Science Academy Young Scientist Award and the prestigious Ramanujan Fellowship from the Department of Science and Technology, Government of India. Dr. Vidya has additional awards and honors and numerous publications to her name. She is the founding member of the Indian Society of Evolutionary Biologists, in addition to being a member on different professional societies. We are very happy to have you, Dr. Vidya. Thank you for joining us and adding to our amazing phenomenal line of speakers. We're looking very excited about your talk and looking forward to it.
Thank you so much. Shall I?
Yeah, if you don't mind, you can share your screen.
Can you see my screen?
I can, yes.
In the slideshow mode?
Yes, now I can see in the slideshow mode. Thank you so much.
Good morning. Thank you so much, Sanjana. I'd like to thank Biologically Speaking and BiasWatch India for organizing this meeting and for inviting me today. So today I will be talking about some work that we have done in our lab on the socio-ecology of female Asian elephants. I will quickly tell you about a few different things that we do in our lab. We work on animal behavior largely. So we've been working on Asian elephant sociality, trying to look at a few different things, associations amongst females, males, allomothering, and so on. But today I will talk about associations amongst females. Sorry. We've also been looking at communal roosting in minors as part of the work that we do on animal behavior. In the area of phylogeography, we've been looking at Asian elephant phylogeography, trying to trace different patterns of historical movement to explain the genetic patterns of diversity that we see in Asian elephants today across the range. We've also looked at comparative biogeography, examining 5 different species across the Western Ghats and trying to see how the Palghat Gap has shaped their genetic structure. In the area of conservation biology, we work on elephants and road traffic, how elephants respond to road traffic. We've used agent-based simulations to look at some census techniques, and we use non-invasive sampling as well as molecular sexing and genotyping to census small populations. And finally, in the area of evolution, I work collaboratively as part of the Foundations of Genetics and Evolution group to come up with conceptual areas of looking at a framework of inheritance, as well as trying to look at the importance or lack thereof of niche construction. So coming to my talk today, as you know, being social can have a lot of different benefits, such as defense against predation, the improved ability to find food, and cooperative care of offspring. So sociality is thought to have evolved as a response to these different benefits from interacting with conspecifics. Conspecifics are other members of the same species, as well as conflict from conspecifics and various ecological factors that are present. So trying to find out the relative roles of these ecological factors, as well as the benefits and conflict from conspecifics, forms a central part of a lot of mammalian behavioral studies and also forms part of what we do. So as you know, there are varying degrees of sociality from animals that are solitary to those that show aggregations in which group compositions and sizes can change over short periods of time. And individuals in these groups are random associations of individuals in that area. To animals that are more social, in which group compositions and sizes do not change over the individual's lifetime, with the exception of births and deaths and the occasional dispersal. So in order to examine how ecological factors and individual relationships affect the evolution of different social groupings, we have what is called the socio-ecological theory, which offers us a framework to look at these different factors. So according to one part of socio-ecological theory, we have this framework which suggests that female social groups are formed as a response to predation and resource availability, while male social groups are formed as a response to these as well as to female social groupings. So if we think of an area like this, where the dark green represents high quality resources and the light green represents low quality resources, in an area where we have abundant, uniformly distributed high quality resources, we can expect individuals to be distributed singly and not form groups. The reason why I'm showing monkeys here is because socio-ecological theory has largely been studied in primates. Now, in such a scenario, if we have a predator, we have group formation here. Now, because these groups have been formed only to defend themselves against predation and not because of competition for food, females are free to move between these different groups. That is, they can show dispersal across these different groups. And all the females within a group are equal. So there is an egalitarian society. And we call this the dispersal egalitarian society. Now, on the other hand, if resources are scarce in that they limit female reproductive success. So if we think about this area as opposed to this area on top, here we have the formation of more long-term groupings, which are called female bonded groups. The nature of these female bonded groups depends on how these resources of high quality in dark green are distributed. So if we think of this scenario, these resources in dark green form large patches, which can support entire groups. So one would expect a lot of competition between groups for access to these resource patches, but we would not expect competition within these groups. Therefore, females cannot disperse from their group and join another group because of this high between group competition. Now, because there's no competition within groups, females also are equal within groups. It's an egalitarian society. So we have a resident egalitarian society. Now, on the other hand, if we had these high quality patches being very small, such that individual females can monopolize them, and these patches are clumped together, we have group formation, but there would be a linear dominance hierarchy within a group because dominant females can access all the resources within a small high quality patch. These hierarchies are often based on relatedness or nepotism. Females here again cannot disperse and join another group because of the strong hierarchy within the group, and they would end up at the bottom of a hierarchy if they disperse. So this would lead to a resident nepotistic society. On the other hand, we can also imagine a scenario where there is potential for both between group competition as well as within group competition. In such a case, we expect that the dominant females within a group become more tolerant of the subordinates, and therefore the subordinates have an incentive to remain in the group. If the dominants were not tolerant, they might disperse to another group, which would be disadvantageous in between group contests because their group size can play a role. So here we have a resident nepotistic tolerant society where there is a dominance hierarchy which can be based on nepotism. but the dominant individuals are more tolerant of subordinates. Coming to male societies, males may be found with female groups, either singly or multiply, depending on predation risk, depending on the tolerance of males by the females in the group, depending on the monopolizability of females by males, and so on. In other species, you could also have males living in different societies than females if the kinds of food or amounts of food required by the different sexes are very different. But that's a topic for another day. And today I will talk about female societies. We study elephants because they are highly social, they inhabit ecologically diverse habitats, and they show a combination of traits that are not seen in primates, especially that adult males and females live in very separate societies so that we can study them without interference such as infanticide that you find in many primate societies. So today I will tell you about two questions that we looked at. What kind of social structure is found amongst female Asian elephants? And what is the nature of dominance relationships amongst females? Now, in order to do this, we set up this long-term Kabini elephant project in Nagarhole and Bandipur in Karnataka, which have high population densities of elephants. Nagarhole and Bandipur lie in the Nilgiris Eastern Ghats landscape in southern India. They are separated by the Kabini River, on which a dam was built in the 1970s, and therefore there is this large reservoir, which in the dry season opens up to form this grassland, where you have high visibility. Now, we individually identify animals using a combination of various natural physical characteristics, such as ear shape. So one can look at the inner edge of the ear like this. One can look at the ear fold, whether the ear is not folded on top, folded forward completely, or folded backward. We can look at the presence of nicks, tears, and holes in the ear lobe length, the ear length itself, various tail characteristics, and back shape. We can also approximately age elephants by using their heights. And we can get heights either by measuring the forefoot circumference, which is half the shoulder height, or by using relative heights like this, or absolute heights by shining calibrated parallel lights on the animal and taking photos. By doing this, we have identified over 600 adult and subadult males and females, and we follow these animals opportunistically. Now, males disperse from their natal groups when they're about 10 years of age. So today we are looking at the female society. We knew that females have some kind of fission-fusion society. So what is a fission-fusion society? A fission-fusion society is one in which the group sizes and compositions vary across short periods of time. But unlike the aggregation which I told you about, groups here are not just random collections of individuals from the area. Fission-fusion societies are thought to have evolved in response to spatio-temporally varying resources, such that if resources are high, you have multiple individuals grouping together. So this thick black circle represents a group, which is a set of individuals we see in the field. And each of these circles here is an individual adult female. If resources are low, these groups can split up into smaller groups, and they can fuse into a larger group again if resources are high. These fission-fusion societies are seen in a variety of species shown here, including humans. Now, in a fission-fusion society, a group is basically a set of individuals that we see together in the field, which show coordinated movement or affiliative behavior. And a clan is the most inclusive social unit. So all the individuals of a clan will usually not be seen together in a fission-fusion society. So for instance, here we have 3 different groups, all belonging to the same clan. But you can see that they are of different group sizes and compositions. So what we do is we go out to the field and we record the animals that we see. So we have the different names of animals and we see them associating together. Then we can construct social networks based on who is associating with whom. We can look at these associations in terms of an association index, which is basically the number of times 2 individuals A and B are seen together, divided by the total number of times they are seen. So AI can vary from 0 to 1, 0 indicating no association and 1 indicating complete association. So using these for pairs of individuals, and based on 6 years of data, we constructed this social network for 330 adult females. Here, every circle is an adult female. Every line here, every edge in this network is an association between these 2 adult females. And you can straight away see that this network is highly non-random. This network is partitioned into a series of communities. And we can partition this into communities based on a statistic called modularity, where basically we try to ensure that most of the connections are within a community compared to across different communities. So these different communities are differently color coded here. And every color here represents a most inclusive level of social grouping, which is the clan. So if we think about this clan, some individuals of this clan may have been seen together as a single group in the field at some point in time. We may have seen another group comprising these 5 individuals. We may have seen another group comprising these 6 individuals and so on. But one would have to do this network analysis to find out what the composition of the entire clan is. And therefore, we find that this clan is the most inclusive level of social organization. We can also look at what happens within these clans and find out whether all the individuals within the clan are somewhat similarly connected to each other, or whether there are substructuring within clans. So for instance, here, these sets of individuals are more connected to each other than to these other sets. So this would mean that this lighter green circle represents a first level community within the clan. And this darker green circle includes the entire clan. So if we had this kind of substructuring, this would be called a multi-level society. While if we did not have that, it would be called an individual-based society. So what we found from our data was that some clans, the smaller clans, had an individual-based society, and the larger clans had multi-level societies. Now, what we wanted to do is to find out whether individuals that associate together are also more related to each other or not. So we collected dung samples from identified females on observed defecation, and we extracted DNA from this dung and genotyped them at multiple microsatellite loci in our lab. What we found was that the average relatedness between adult females within those first level communities in the clan was about 0.2, which is quite high. It would suggest it's a combination of mother-daughters, full sisters, half-sisters, and more distant relationships. So mother-daughters and full sisters would have a relatedness of 0.5 and half-sisters of 0.25 and so on average. We also looked at the top associate, that is who did a female associate with the most, and what was the relatedness with that female. So the average relatedness to the top associate was 0.25. The average relatedness to the second associate was 0.14, and so on. So the top and second associates were more related than the average associate. And this also tells us that the associations are biased towards relatives. The average relatedness between adult females within clans was higher than 0, which means that overall these clans were related to each other. Sorry, the adult females within clans were related to each other, but this was not necessarily true of all the clans. Now, coming to the second question about the nature of dominance relationships, why we were interested in this was that there are 3 different predictions about what kind of dominance relationship should be seen in Asian elephants. First, we know that elephants are bulk foragers. That is, they have to eat for many hours every day and eat a lot of food. And therefore, one might expect that they are not very picky at what they eat. And because such food might be distributed very widely, one might think that they show an egalitarian society. On the other hand, because we found that clans had related females, there is a potential for nepotistic dominance hierarchy. So one might expect a resident nepotistic society. Third, in the African savanna elephant, which is the closest relative phylogenetically, along with the forest elephant, an age size-based hierarchy was seen. And therefore, it was possible that such a hierarchy would be found in the Asian elephant also. We therefore carried out behavioral sampling to look for various behaviors such as chasing, charging, pushing, shoving, and so on, which are dominance behaviors. And we found that there were clear winners in almost all the interactions which took place within clans. But the number of interactions which took place within clans was not very large. Now, to find out whether there's a linear dominance hierarchy or not, we need to see if there is transitivity in interactions and whether there is unidirectionality. So if this is a dominance network where each of these circles is an adult female, and this arrow points in the direction of dominance, if A is dominant over B, B is dominant over C, and in turn, therefore, A is dominant over C, this would be a transitive relationship. While if A is dominant over B, B over C, and C over A, that would be a circular relationship. So if there is transitivity, we would get a dominance, a linear dominance hierarchy. If A is dominant over B, always, that would be a unidirectional relationship, as opposed to B also retaliating against A, which would be bidirectional. What we found within clans was that there was no formal linear dominance hierarchy, but this was not so much because of circularity as because most individuals did not show any dominance interactions towards each other. We found, though, that there was unidirectionality almost always, and therefore there is some sort of orderliness in the dominance. Moreover, initiators tended to be older than recipients. Therefore, this orderliness in dominance was based on age. When we looked at between clan dominance, we found that the dominance interactions between clans was much higher than those within clans. And these behaviors that were shown were also of much higher intensity compared to other dominance interactions seen and qualitatively with other populations. For instance, here we have a between clan dominance interaction where there are females from two clans engaging in head-on fights, which we usually do not associate with female elephants. We see this more commonly with males. We considered a clan to have won this dominance interaction if it was able to displace the other clan from that particular feeding area. And what we found was that in such between clan dominance interactions, there was a clear winner in only about half these interactions. And there was an effect of group size on winning, which is also what we had expected from the socio-ecological theory. We found that the winning group, when there was a clear winner, the winning group had a larger group size overall and also a larger number of adult females than the losing group. Now, in order to find out whether this was expected by socio-ecological theory, we would have to look at resource availability. So we estimated the tree, shrub, and grass biomass in different transects and plots across the Nagarhole National Park. We also looked at grass biomass in various vegetation plots along the Kabani backwaters. We found that the grass biomass was much higher around the backwaters than inside the forest. And grass forms a very large component of what elephants eat. So basically, we had this kind of a scenario where there was a large resource patch and not this kind of small patch of high quality, which single individuals could defend. So the Kabani backwaters itself seems to be a large resource patch, which would explain the high between clan competition. So if I go back to the original schematic, we know that elephants don't show a dispersal egalitarian society because we know that adults do not have any natural predators and females do not disperse across these different groups. We found instead that associations were non-random and there were these different kinds of fission-fusion dynamics, an individual-based society or a multi-level society. We found very low within clan dominance, which would therefore rule out the nepotistic society in which most of the dominance happens within the group, or in this case, within a clan. Instead, we found an effect of age on winning. Therefore, we can also rule out an egalitarian society because there was some orderliness to dominance relationships. We found high between clan dominance compared to within clan dominance, which suggests a tolerant society. And we still have to look at the effect of nepotism on this between clan dominance. We therefore have a situation where between clan dominance rather than within clan dominance seems to be driving the social organization of elephants in this area. And it is possible that this is an effect of the Kabani Reservoir, which was formed only in the 1970s. And maybe elephants are reacting to this in the form of dominance for access to a large resource patch. In order to find out whether this is more generally applicable, one would also have to look at the ranges of other elephants which do not share this resource. So I'd like to thank the funding agencies, DST, National Geographic, CSIR, and JNC for funds and support, the State Forest Department of Karnataka for research permits, Dr. Ravi Kumar and Dr. Poddar for identifying plant specimens, The work on social organization was done by Nandini for her PhD thesis. Kirti Priya helped with this, although her PhD thesis was on male associations. Hansraj looked at the dominance relationships between groups and within groups as part of his PhD thesis. And Revati has been looking at allomothering. Various people have helped with fieldwork. And I thank you for your attention. Thank you.
Thank you so much, Dr. Vidya. Everyone, yeah, let's give Dr. Vidya a round of applause. And as I said, this is what we have in our virtual background. I'm sorry, in the virtual environment. So we do have a few questions. So
Sure.
yeah, we have the first question is from Sonali Sengupta. She's asking, do Asian female elephants, which are genetically related, use smell more predominantly as opposed to vision for in-group selection or choice?
So in general, elephants use olfaction rather than vision. So their sense of smell is very highly developed. Their vision not so much. So that is generally true.
Yeah. Okay. Thank you. Another we have from Manish Mandotra, who asks about the methodologies. What are the methodologies adopted after extraction of DNA from fecal matter, both computational methods and wet lab methods?
Okay, so we do PCRs to, we have already standardized some microsatellite loci. So we do PCRs to amplify those microsatellite loci, and we run it on a sequencer, and we score it with Gene Mapper software, if that's what she wants to know.
Okay. Hopefully the question was answered. Sonali Sengupta has another question. Firstly, she says, great talk. Thank you. So is there greater genetic relatedness between members of an in-group as compared to an out-group?
Sorry, is there greater relatedness between members of?
Of an in-group as compared to an out-group?
Yes, by definition, but yes.
Okay.
Yeah.
And Shawnchari Gorai asks, we know that the herd of elephants come to the human locality searching for food. Then is it called clans? I think you've covered a bit in your talk.
No, so when animals come to search for food, that does not necessarily have to be a clan. Often males are seen dispersing and therefore tend to come into conflict with humans more frequently. And they can be from different clans because males disperse from their native clans.
Okay.
And also in disturbed areas, we do not expect this clan structure to be maintained so well. For instance, we've done some work in Vietnam after the war because elephants were highly decimated. If you look at the elephants that are remaining, you have remnants of different groups that have come together and appear to be a group now, but they actually have different mitochondrial lineages.
Okay. So yes, her follow-up question was, if yes, then which type of clan, individual or multi-order?
Okay, so nobody else has really looked at this individual-based and multi-level society before in other populations. So we don't really have a comparison for other populations.
Okay.
Yeah, we need many more studies of individually identified elephants being followed over long time periods. We don't have very many.
Okay. And another question, if we can take, do females give clans decide on migratory routes? I'm not sure. Smrita Chaudhary, could you frame the question differently? She's asking, do females give clans decide on migratory routes?
So usually it was thought, at least in African savanna elephants, we know that the adult females, which are the oldest, which are called the matriarchs, are the ones that lead these females across different areas. So if they have to migrate from one area to another, that would be done by the oldest adult female. But we know from our work that the oldest adult female is not as crucial in our population or at least doesn't seem to be in day-to-day situations. Of course, oldest adult females could be a repository of information and could be very crucial over the long term. But in day-to-day activities, we do not find the matriarch playing such a big role in Asian elephants compared to the African savanna elephant.
Okay. Thank you. And Smrita, thank you for clarifying the question. We do have a few more questions, but maybe people can email you later or get in touch with you. So thank you everyone for the questions, and we apologize we can't take all the questions. And thank you, Dr. Vidya, for your work.
Thank you so much.
And also it's so interesting to know like the dynamics, and also in the present context of climate change, habitat destruction. So it's very
thank you so much.
Yes, thank you. We'd like for you to join the remaining conference if you can, but if you have commitments, we totally understand.
Sure.
Thank you so much.
Thank you so much.
Thank you. So let's give Dr. Vidya another round of applause. And with that, we end this session, the Jagannathan Vijaya session for today. We do have another speaker in this session, which is Dr. Uma Ramakrishnan, whose talk, Dr. Ramakrishnan's talk is tomorrow at 3 p.m. So do join in for the talk. And today, so we'll move on to the next session, which is our Shakuntala Devi session on computational biology, for which I will hand it over to Krishnamurthy Srinivasan, who is a graduate student at Indian Institute of Chemical Biology. Over to you, Krishnamurthy.
Thank you. Welcome everyone to the Shakuntala Devi session on computational biology. Shakuntala Devi, as we all know, is an Indian mathematician, a writer, and popularly referred to as the human computer for her superhuman mathematical ability. She was born in Bangalore, and her father, a circus worker, recognized her remarkable ability to memorize numbers when she was a kid of 3 years. And then he used to take Shakuntala on road shows to display her amazing abilities. Her arithmetic abilities, without much formal education, really amazed people as she traveled all over the world displaying her skills. Her skills were tested at the University of California, Berkeley, where she famously completed calculations of 7th root of 9-digit numbers and the cube roots of 8-digit numbers before even someone could note it down. She even outdid a computer, the UNIVAC 1101 computer, state-of-the-art computer in her time, in complex calculations, which fondly gave her the nickname, the human computer. Apart from that, she was a prominent author who has written several books on mathematics, puzzles, and has also published a book on the society's understanding of homosexuality in India, and a work which is considered to be pioneering in her field. So, in honor of India's beloved mental calculator, Shakuntala Devi, biologically speaking, is proud to present to you the Shakuntala Devi session on computational biology, featuring Dr. Lipi Thural, and Dr. Sucheta Tripathi. Now, Dr. Lipi Thural is a scientist at CSR IGIB in Delhi. She did her bachelor's in biotechnology at Amity University. Juggling regular college classes with her programming classes in the evening, she improved on her skill sets as she decided to enter the world of computational biology. She completed a master's in bioinformatics at the Banastra University in Rajasthan. From there, she did her PhD in the University of Heidelberg, Germany, with Professor Jeremy C. Smith and Dr. Isabella Diarni from University of L'Aquila, Italy. She returned to India in 2012 as a DST Inspire Faculty Fellow at CSR Institute of Genomics and Integrative Biology, New Delhi. Since 2016, she is a senior scientist at IGIB. Lipi uses diverse computational methods combined with high-performance computing to investigate dynamics of biomolecules with particular emphasis on the interaction of proteins with membranes. The methodology used relies on strong biophysical and statistical mechanical principles to investigate structural changes underlying biological phenomena, which are crucial to understand the function and mechanism of cellular processes. The long-term goals of her research are to trace the origin of diseases, to discover new therapeutics, and develop bio-inspired engineering system. With that, we invite Dr. Lipi to take over from here. Over to you, ma'am.
Thank you. Thanks a lot for that introduction. Can you hear me?
Yes,
I
yes.
hope. Okay, perfect. All right. So I have a list of people to thank because I'm quite impressed with the way you all have conducted this series. So I really want to congratulate the organizers and the team that is behind Biologically Speaking group. It's tremendous that what you guys have put together and also very professionally put through. So I'm really impressed. So thank you for giving me this opportunity to speak in front of this very wide audience. And I also want to thank Bias Watch India to sponsor your event, which is definitely to support these young group of scientists. So yeah, I'm particularly very excited as I've been introduced here as a computational biologist in the team of Shakuntala Devi, which is quite yeah, admiring personality. And we've all seen her movie also, which has been lately made. So I'm quite thrilled, to be honest, in this session that we're all having lived under these very famous personalities. But the idea of computational biology now has evolved a lot. And I'm going to present to you some of our work in the beginning, but also give you a very general idea about that what I consider that every biologist should now learn a little bit about biocomputing, which is now going to become essential. And I'm now sharing my screen for the presentation. Can you see this screen?
Yes, yes. Yeah.
All right. Okay. So I'm going to present reasons why basic computing is essential for biologists. And with the start of some of the background of our work, which we do at IGIB. And the idea is definitely that biology covers length of different scales. And it starts, of course, from the human body at very large resolution, which is studied clinically and phenotypically in various forms. We have various model systems from Drosophila to mouse to study the impact of several different conditions, different environments, such that we mimic the processes at human physiological scale. What is interesting is that there are these cellular systems that are also available at micrometer length scale, and these also reasonably capture the individual cellular complexity. Now, coming to what we know in nanometer scale, are these viruses, exosomes, large bodies which are encapsulated with some sort of membrane from outside and have a complex and intricate protein membrane interaction continuously from the environment. And these are not large, but they're very complex. They are in the range of 20 to 200 nanometer. Of course, this can vary for other organelles. And within them are these host of machineries that we all know as proteins, ranging in few nanometers, and DNA, which is also the essence of all the biomolecules. But what constitutes these proteins and DNA as an element are, of course, the atoms. The atoms are the basic element of all the living systems, and how they come together essentially define how we are functioning. So this is what has fascinated me in science to come and work in the field of atomistic structures, and they really fascinate me that how do these structures continuously adapt with different surroundings. We do not know yet that how most of them, we do not know that how do they connect to the phenotypes, but that's the idea, right? That if an atom is moving, how do we sense? For some of the molecules it does, and we do know. And the idea is to cover that area in much more depth in coming years or decades. So at atomistic time scale or atomistic scale, we recognize that ligands or small molecules bind to these proteins and they inhibit or activate certain processes. Now, the complexity is when they are at the heterogeneous scale, meaning that these biomolecules are not just of same kind, but for example, of different kinds, like lipids interacting with small molecules, and those small molecules are then sitting on another complex on a membrane. So this is where we've started to capture these heterogeneities at atomistic time scale. And what other things constitute these are not just so just highlighting here those heterogeneities. Protein interactions, protein small molecule interactions, small molecule membrane interactions, as I just showed, but then also protein embedded on a membrane. And on the right hand side, you see that complex model of interactions with a lot of intricacies going on. So um, my lab focuses on this and much more in these three areas. And one is primarily in silico autophagy. So I'm going to elaborate a little bit more on that. There is a process of autophagy where there is a complete recycling of cellular materials, and we are interested to mimic this process or reconstitute the process in silico. And we're also interested in dynamics of membrane signaling proteins. And GPCRs have been also a target in our lab in the past few years, majorly driven by one of my postdoc, Nidhi. She is quite keen on working on these membrane signaling proteins. And then there's one PhD student of mine, Surbi, who's working on Notch signaling proteins. So we're really interested in how these large assemblies can now talk to other cells. And then lastly, of course, being computational biologists, how to continuously develop new computational platform or utilize the existing ones and adapt to our needs. And with this, I will come to the focus area of my research where most of my PhD students are working. That's the autophagy pathway. The LC3 is in focus. The LC3 molecule is something like a biomarker in this particular process. The overall process or overall progression of autophagy can be seen from the building up of an isolation membrane, which is there in the cell, to elongation and completion to complete formation of this autophagosome vesicle. This vesicle encapsulates cellular cargo. Now, this cellular cargo can be really bulk stuff which is not needed for the cell, which is called macroautophagy, and the other targeted vesicles like targeted organelle like mitochondria can also be degraded, which is then called mitophagy. So there's a lot of specifics involved in this process. We are going to focus currently on the macroautophagy, which is the degradation of cellular waste in general. And here is where these molecules LC3 are available, and they are sitting on these membranes to help in multiple things. One, elongation of the membrane. Secondly, there is a form of LC3 um, which is distinguishing whether autophagy has progressed or not, meaning the LC3 cytosolic form is not on the membranes and hence not seen as a characteristic feature of autophagy, while there is a lipid anchor protein which comes as a separate band on the western blot, which is attached to a lipid called, covalently attached to a lipid called phosphodiethanolamine. And that particular LC3PE, when seen on a blot, is known to be a marker for autophagy. And this is how the complete progression is measured because we can see punctars on the any live cellular imaging platforms for the presence of these LC3. Now, this is quite remarkable for us because at the atomistic scale, we can now see these two things or these two protein entities very differently. So we sort of started simulating at very initial time when I joined here at IJV, and we do molecular dynamics simulations. So anyone who doesn't know about a large things about molecular dynamics simulations, they are based on Newtonian equation of motion, and they capture essentially the time-dependent characteristics or dynamics of a particular protein. And this is based on something what we call as a force field. Okay, these are empirically calculated parameters. And for example, the fields that we know now that go into these force fields are something of bond angles, bond lengths, of carbon, nitrogen, of these certain number of parameters that define what is it to be called a protein or a DNA. Once we put these parameters, we can essentially simulate them. We can carry out a large scale. So there is a time dependent trajectory. We need stack of laptops, which is in other words supercomputers to do such simulations. Okay, so I'm showing you an example of an MT simulation, that is molecular dynamic simulation of a protein called LC3. The protein has a PE chain, which is shown in the red color. So if I move very slowly, then you can see that the protein has a twisted PE chain, but once it comes on the membrane, it has a very specific way. Now just watch. It has a very specific way in which this phosphodiethylamine chain will enter the membrane. So there's one chain which enters, and then it is followed by another chain that enters the membrane. Now this entire complex is mimicking those LC3 molecules on the autophagosomal membrane. And now we can ask plethora of questions because theoretically we have observed the phenomena, and this took us a very long computation time, although this was coarse grain simulation, so a little bit into technicalities. This is a little bit of coarse graining here, but we could still manage. Now we can ask the questions that what are the residues that are critical for this membrane interactions? And we did, of course, firstly, multiple simulations to be statistically sure of the regions that we were proposing. So we did not just one experiment, but 15 of these experiments. And the blue color shows the time scale at which the protein entered into the membrane. So this is sort of stochastically driven. And we can see that nice stochasticity in these 15 simulations that at some point it entered at some region and at some point because they all have been given different velocities. So once you find this large conformational ensemble of LC3 dynamics, we can go to the experimentalist who we collaborate with to ask the question that can you now check which are these residues and can we now sort of modulate the process? And that was really interesting because LC3 wild type, as I told you just before, it has a lot of punctars. And you can see these punctars here. And now we started to give the mutations one by one, series of mutations around the region, which we called as from 65 to 69 region residues. And we gave one after the other. And we could see that there was some changes in 65, 68, 69, and there was complete loss at one particular mutant. And this almost really confirmed our observation, our hypothesis, our computational finding, the predictions that there are these group of residues that when mutated does not let the LC3 bind to the membrane. And this was really interesting for us and also kind of really stimulated a lot of LC3 driven, autophagy driven work. And this also gives you a summary of what we are doing. And now I'm going to use a lot of our data, but I'm going to put things in context with the broad general title that I have made for this seminar. That is that why biocomputing is becoming essential for everyone. So one group is definitely working on these extensive simulations. You need background, you need training to do these simulations. Um, in this particular seminar, I'm also stressing on the fact that there is a biocomputing element in all of our works and why it will help us. So I'm not going to give you boring details that learn Python, learn MATLAB, learn these things which you're going to get it everywhere. I'm going to give you a perspective on how it will help you if you are a PhD student or a scholar who's working in the field of experimental biology. First and foremost, uh, it's something eminent that data is massive and it's increasing every day. So meaning that I've made this graph when I started as a PhD student, and a few years back, I made it just to see what things were happening. And I can tell you because I pasted this old graph to showcase even in the 4 or 5 years back when I made this, there have been tremendous progress. For example, the number of genomes when I started were few hundreds, and there were 16K. And now we all know that human genomes are 100Ks, and there are a lot of genomes that have now come up, both naturally occurring variants capturing naturally occurring variants as well as the disease variants. Protein structure were only few thousands, tens of thousands, but now they have crossed 150K and over that. So now we also have these ensembles, and I'm going to show you our recent work in SARS-CoV-2 spike protein, how we incorporated this large sum of information as well. And of course, the supercomputing performance, needless to say, is in petaflops now, is in petaflops. And when I started, it was no one knew what is petaflop computing. And of course, peaks of supercomputer elsewhere also have now are aiming for much more. What has also happened, if you see in the below panel, is that people now, general public, are interested in monitoring their parameters, health parameters. And this was not there, this was zero a decade back. Now there are people who are interested in wearing fitness devices that is collecting data. And this data will not just collect noisy data in future, but will also collect precise blood-based markers. What will you do when you have so much of data, right? So that really is very interesting thing. So you have to think about it, that when this data is going to come, then how is the whole scenario of biology going to change? So this was one uh, particular thing, and this has led to growing information per gene or per protein, meaning that if you study a particular network, okay, and that network has, let's say, X number of genes, and X number of genes, you are simply making the size bigger depending on what all information you know. So here, at present, we are in the situation where there are few proteins that are relatively studied more, and the few are less. But the point I'm trying to make here is that it's going to be tremendously drastic in years to come. So every node, every gene, every protein is having a lot of information. So what is the example I can give you? The example that illustrates all these increased data hubs allowing greater weights for specific protein. So right now you have these missing functional connectivity, but the examples that really exhibits this data in a huge way, sorry, I have an animation, is the spike protein. Okay, this is an animation that I made, sorry. Now, just see this. So the work captured mutations that are accumulating on spike protein. And through this, we tracked 3 lakh genomes for a period of 1 year. We saw 2,500 mutations, and we could now track the genomes across time to get mutational ensemble. But after mutational ensemble, we could also now get 50 odd structures of spike protein with different conformations. So these conformations allowed us to now understand both the genomic variations as well as conformational states. And this is not available, this much of data was not available for any other protein. But just right in front of us, because of the rush, because of the progress in the pandemic, we can now see how measurables have increased dramatically. Not just one structure, 50 odd structures. So you have not just one functional state. These are all experimental structures. So if anyone is questioning about, the prediction power, etc., I'm just stressing here, these are all computational, these are all experimental structures. Okay, and then we had 3 lakh genomes. So 3 lakh genome is a very large number to be monitored and to be addressed. And through this, now we already know so many variants of concerns, so people could now adapt and do so many different things. So this was really interesting work where we saw the progress of data points increasing every day. So this was my first point. And now the second is, of course, visualization of data. And this doesn't concern only computational biologists, it's for everyone now. That how can we make sense of these meshy data? This is a word that we also published a few years back. But this network is showing several proteins that are involved in apoptosis or DNA repair or different pathways. How do we make sense of it? And for this, if you can learn little bit programming, not just to write programs, but to clear data points, this will help you to visualize. For example, there was a work that we did in autophagy, which is now published in Autophagy Journal. We tried to classify, and on the left-hand side, you can now see LC3 proteins, and these are LC3 homologs in humans. And you have ATG8, which is in yeast. And on the right-hand side, you have all the organisms. So with simple visualization, you can now see how many of organisms have that particular protein. For example, ATG8 is there in maize, in yeast, in honeybee, in Drosophila. So these all organisms have one ATG8 protein. Okay, and this one is also listed here. The rest of them have a range of proteins in this family, ranging from 4, 5, 6, 7, in fact, 8. We have 8 proteins in zebrafish. And with a click of this particular Sarko's plot, you could see what is the dimensionality, where to focus on. And I don't think this is just left to computational biologists. You can also make these plots which simple knowledge of algorithms, which is my next point. Yeah, so this is a similar phylogenetic tree where you could now club according to the functions, and you could now see that what we proposed in this work was that LC3 is largely function-specific phylogeny. Um, the, the other point is the analytical skills. So the tools allow you with better analytical skills, meaning that if you are measuring certain property as a function of time, what would you do? So you would have not just one particular structure, not just one particular access to interaction, but just multitude of it. So here is where I would like to introduce this Jugado programming skill concept, where we all used to in India, that if you can make function little bit of clearance of data with what we call as data handling, that you could really now extract from the files particular questions, and asking those relevant particular questions will allow you, or will at least give you an analytical mindset to dissect your research problem. And that I think is really beautiful. And the second last, and I think this is also important, that as computational biologists, we work on very complex algorithms. But these algorithms allow the entry of new hypotheses. For example, here we have taken complex membranes from endoplasmic reticulum, Golgi apparatus, mitochondria with different diverse lipids. And once we take them in these constituent lipids, you can now measure forces. And this work is relevant for membrane mechanics. There are a lot of uh, processes which involve mechanical change of membranes. So we wanted to measure that if individual lipids can be discerned to understand this mechanics. I'm running short of time, so I'll have to go fast, I guess. So this particular process of understanding the algorithms understanding a bit of computing allows you to also understand the algorithms um, for your own work. So can you now understand a little bit about how to take the work forward? And I think this is also interesting because if you're in the field of transcription factor binding sites or any other prediction that you routinely do, if you understand that algorithm better, you'll be able to handle your data better. And last, not the least, is the integrative knowledge, which this whole biocomputing brings. Because there were eras when we were trying to understand what goes where, which is that um, the complete idea of human genomes, that who is the important question. And then we are currently in this era where we're trying to just say where it goes. Can we now monitor that using so many tools like confocal, EM, and the protein localization and the activation upon the cellular queue is the current question. But this is all technique based, one, you see, and techniques are not limited. They will always evolve. So the era which I think is going to be really critical is that how do these things achieve functional specificity? What are the drivers of functional specificity? And this will involve us to decompose or rather decode a large amount of data. And for this is what, if we understand all this together, we will obtain a better integrative knowledge of the biological process or the protein that we are studying. So with this, I want to now acknowledge my crazy team, which I'm super proud of. And there's a super crazy picture here as well with the team. Some of them have left. Arjun Nidhi is on her way out. Asad has left. And in this picture, we have Surji, Dipanshi, and Saman, who are an early group of PhD students. And I have new group of PhDs, Akansha and Shahnu, who have recently joined, but are not on this picture. And of course, my collaborators, funding agencies, and thank you again to all of you for listening. Yeah.
Thank you so much. We have a lot of questions. To start off, Sundaram Acharya, who's also a member of our group, he was asking that he was wondering what is the scope of your work in designing peptides for therapeutic purposes.
Yeah. So I think designing peptides has been in the in the research domain for quite a while. For example, there have been already successful therapeutic peptide design in the case of antimicrobial. The creams that we get, they are mostly peptide based and they target several of these microorganisms because they can target their membranes. And a little bit around that, we've also worked in antimicrobial space. So there's a lot of potential because the peptides have the unique ability to function in various ways. For example, you can have amphipathic helices that have just series of charged amino acids on one side, and that allows them to penetrate the membrane very easily. So these properties of peptides can then be exploited for the entry of the peptides or carrying any kind of cargo or any kind of biological nanopeptides into the membrane. So they have been really utilized. And now with the era of so many new techniques, they have now been looked at in very different light as well. But it's a very fascinating area.
Smrita Chaudhary is asking that in the event that wet lab experiment does not corroborate a computational hypothesis, how should the situation be dealt with?
It should be dealt with honesty. And, I agree most of the time we are into the business of model fitting, that how do we fit different parameters? I really love this question because I would like to, start with unbiased results where we see what we see and we go to the experimentalist to say, this is what we see. Sometimes these things do not match. And I think it shouldn't be a complete stall of research as well. One should ask further questions. For example, sometimes I've noticed that there are things to be explored. For example, if you're looking at an interaction and you've not found an interaction in atomistic detail, but you're finding an experiment. So could you now ask further questions? Can you change certain conditions? Or you could ask this. So it should be not a yes or no sort of a mix and match. It should be a continuous evolving research question that should allow you to study things in depth. Even then, if it's not working, it's not working. That's the thing we should report, that the atomistic picture or the other experiments we do not observe it. So it's fine. But I think it should be a continuous progression of events or different conditions which can be checked.
Manish Malhotra asks, can you please mention the reasons behind the lack of algorithm understanding, and what is the way out for this?
Yeah, I also realized when I was making that point that so the large part, and I have to confess, we are all a part of it, is the lack of our ability to communicate mm, to the not the end user, but also general users. So what happens is that we tend to focus on who's going to use our tool next, which is going to be mostly computational biology field, which then evolves on it and works on it. But there are only few, which is obviously everyone knows those web-based tools that everyone can use. But it's inability of the people who make the tool to communicate it better, to make the algorithms, and it takes a long time as well. So what happens is that if we are making an algorithm in a particular program, and if you write to us, we make that available. But to make it in an online mode, not all the algorithms can be worked there. So I think that's the major reason that it gets stalled and doesn't reach the wider audience. But it should be done nevertheless. And I think it just made me this part also realize that some of these things end a little bit short, but also they are very difficult to make it in an online mode because you require certain kind of input files, certain kind of output files. So either everyone should make an effort to make that, which is difficult because it requires enormous amount of computing infrastructure, enormous or different kind of computing expertise as well. So that's the challenge I think where we are dealing with.
Shanchari Gorai is telling us that your talk makes him interested in computational biology. So he wants to know if you can provide an information about some computational biology course for a biology student.
I think I'm quite clear that we have entered the era where you can learn things on your own, and YouTube is the best source. Gone are the days, when I used to write to a trainee student that these are the links, this is the attached PDF, this is the book, please read this chapter on this subject to gain more knowledge and then come to my lab. No, now there are two links that are available to you. One is YouTube. Write keywords. The more you will know, the more better you get to write keywords. Because right now you can write computational biology, then later you can write conformational ensemble, then later you can write mutational variations, then later you can write, I don't know, force directed graphs. So the more you know, the more your knowledge of the keywords and to search something specific will increase, and therefore mix it with Google Scholar literature. Beyond this, you don't need anything else now. You don't need very classical book knowledge. You are in a very nicely, the era is really interesting where you can learn things. People are learning guitar on their own on YouTube. So I don't think there's anything else which is left.
Thank you so much. It was a wonderful lecture. Thank you so much for
Thank
spending your
you.
time, uh,
Thank you.
despite being on a vacation. Thank you. Means a lot to us.
Thank you. Thank you.
So next, with this, we close our morning session. But we have another speaker in our Shakuntala Devi session on computational biology, who is Dr. Shuchita Tripathi. She will be speaking today at 4:30 p.m. At this moment, we break for our lunch. So see you all back at 2 o'clock, where we'll start with the Dorothy Hodgkin session on structural biology. So thank you. Thanks a lot for tuning in. See you then.